everyone, and Happy New Year. It's a pleasure to welcome everybody for this year's first in the series of First Friday CME series by Indian Association of Biological Psychiatry. It's been my honor and privilege to be the General Secretary who initiated this series. And on behalf of Dr. UC Garg, our President, I welcome you all and take a huge amount of pride inviting Dr. Aleem Siddiqui, who's I think he's known to everybody for various talents, not to mention histrionics. Having said that, he's also the editor of our upcoming journal of IABP. And with that, I'm going to hand over the podium to him to let us know more about this curated access series that we have on. And Dr. Aleem, it's over to you.
Thank you, Ruksheda. Absolute pleasure to introduce this seminar. My good friend Bhaskar is there. Let me introduce the chairpersons. Dr. Smita Deshpande. We all know her too well. She's currently in Bangalore. She's Professor of Psychiatry Research at St. John's National Academy of Medical Sciences at Bangalore. And who does not know Dr. Vihang Vahia? Vahia sir is a consultant psychiatrist. He's known for his quick wits. He's at Breach Candy and Lilavati hospitals. So this series is going on very well. My best wishes to you. What I would request if we can get some articles out of this series for our journal also. Our journal is now live. Website is live. So I'll just post the link here also. So thank you all of you for joining. Welcome, and over to chairpersons for further proceedings.
Thank you so much, Ruksida and Aleem, for that nice introduction. And I think this Friday series by the IABP has really broken new ground in the sense that people came to know about biological aspects of psychiatry on one topic in depth. And I'm hoping that this will continue for a long time to come. And in that same series, we have a distinguished speaker today, Bhaskar Mukherjee, who will speak on epigenetics. Dr. Vihang Vahia will, of course, introduce him because they are great pals, I think. But just to introduce epigenetics as such, which I'm sure Bhaskar will do very well. But taking my role as a chairperson a little bit more seriously, epi literally means on top of. So epigenetics means something that is on top of both genetics and environment. So how does environment in a live and continuous way affect the way our genes express themselves? So this is, if you like, more of physiology than anatomy. When we looked at just the structure of the DNA, it was more of a sort of an anatomical exercise, what existed where. But here it is the living organism. How do things around us, for instance, the air, the water, the experiences that we get as we grow up, how do they influence our genome so that we become different from the others, and perhaps even generations down the line, our generations become different from other generations. So I'll of course wait for Bhaskar to throw more light on that topic, and I'll hand over now to Dr. Vahia to introduce Bhaskar. Dr. Vahia, you are, I think, muted probably.
Hi. Earlier in my career, I learned a small phrase which says, weaker the speaker, longer the introduction. And going by this dictum, all I have to say is that it's a privilege to present to you Dr. Bhaskar Mukherjee. And anyone in this group who doesn't know Dr. Bhaskar Mukherjee may please quit the session. Dr. Bhaskar, over to you. Dr. Bhaskar, you are muted now.
Yes. I have unmuted myself. Thank you, sir, for the short introduction. Introduction is actually, in my opinion, a waste of time. So let's get straight to topic. I would try to show what is epigenetics. I am not going to details of it because epigenetics in psychiatry would take a few months long CME rather than just a 1 hour discussion. But I would show some salient point and I would try to break a stereotype where it is seen as epigenetics. Whenever we talk about epigenetics, we talk about an inert body, an inert DNA on which environment is doing its tricks. We call it passive epigenetics. But epigenetics is an active thing. Active thing means here rest of the genome, that part which doesn't code for protein, it allows various environmental changes to affect gene or not, to affect protein production or not, and various things. It is called active epigenetics. And here I would try to show how these different two things differ and coexist in psychiatry. With this introduction, let me go to the presentation. Please make me co-host.
Amita?
Amita, please make me co-host. Yes, thank you. I have Is it visible?
Yes, Dr.
Is my slide visible?
Yes, Dr. Bhaskar,
Yes.
you are visible.
Yes. So I have used PTSD as a primer because by all means, PTSD is quite essential way of showing how environments interact with DNA in producing epigenetic change. So I would use PTSD as a primer and I would show how epigenetics work in psychiatry. This would be the presentation flow. First, we would talk about schematics of epigenetics, then how epigenetics work in a given gene environment interaction, epigenetics of PTSD, and how many ways we have to assess the epigenetic changes, and then limitation of the present research. So, schematics of epigenetics. Epigenetics or epigenomics is a field of study focused on changes in DNA that doesn't involve alteration to the underlying sequence. Means it doesn't cause change in DNA structure itself. The DNA letters and the protein that interact with DNA can have chemical modification that changes the degree to which genes are turned off and turned on. Turned off and turned on means either a gene would produce a mRNA or it won't produce, depending on various variables. And also these variables can change how a gene is producing a protein and in between stage. Certain epigenetic modification may be passed on from parent cell to daughter cell during cell division or from one generation to next. The collection of all epigenetic changes in a genome is called epigenome. This is a standard definition. Now, when we discuss about epigenetics, there are some terms we should need to know. You know epigenetic variation, chromatin modification, epigenome. I am not going to details of it. I would talk about AP alleles because AP alleles is something you won't hear every day. AP alleles are actually meiotically heritable allelic differences in chromatin state. Means AP alleles are the epigenetic signature that gets passed on to next generation. They can be pure, which are independent of any genetic information. They can be facilitated, where a genetic difference lead to potential to adopt alternate chromatin state or obligate APL. Obligate means altered chromatin state that is fully dependent on genetic variations. Now, most of our genes are either facilitated or obligated AP alleles. Pure AP alleles actually doesn't exist. But most of the time, psychiatrists focus on pure AP alleles because psychiatrists think that gene and DNA is inert and various environmental factors can work on them at their will, which is not really correct. Pure AP allele very rarely exists. This is the first flaw of psychiatric epigenetics that goes on. Now, what can be the epigenetic modification? Various non-coding RNAs, they are becoming center stage of epigenetic nowadays. Chromatin remodeling, another way. Histone modification, we all know about it. Then DNA methylation and N6 methyladenosine. That wraps the basics of epigenetics for most of people. But sadly, that is not epigenetics. That is actually just the surface or even less than surface. Epigenetics is actually the most abused word in psychiatry along with resilience. Resilience probably goes to be the first. Neuroplasticity and epigenetics would contend for second place. And epigenetics is used as any explanation when speaker doesn't know what is going on. Basic schematics of epigenetics starts from this diagram. DNA replication, then it translates into pre-mRNA, which splices change form from mRNA that got translated into protein, and protein are translated into functional protein. Now, all these states, be it DNA. I am trying to write something on the slide. Is it visible? This writing?
Yes, it is. Yes, it
No,
is.
no, the slide is visible, but not your writing.
Those red marks are
The
visible.
red lines.
Yeah,
Yes,
the red lines
yes.
are visible.
Yes. So this is one area where epigenetics come into play, that DNA transcription. Then the DNA format that is also parts of epigenetics. Then splice and spliceosome formation, again that is part of epigenetic. Then translation, this is also part of epigenetic. Then post-translational modification of protein, that is again part of epigenetics. And each area involves thousands, if not more, of processes. in our DNA, there is actually non-coding DNA and coding DNA. Coding DNA contains the genes we know. Coding DNA is about 1 to 1.5%. Rest non-coding DNA, previously we used to think that it does nothing, and it was named as junk DNA. But fortunately, we are finding more and more treasures inside this junk. And we are now understanding that this part of non-coding DNA is what is regulating epigenetics. It codes for tRNA, ribosomal RNA, cutting and splicing of various part of DNA. It causes transposon assemble, reassemble, retrotransposon activation or deactivation. It causes whole genome rearrangements. It also causes small RNA and source of new exons. And other unknown action means there are various open reading frame. Now, open reading frame is nothing. These are parts of DNA which can be manipulated to turn into new genes. And these new genes can give rise to new proteins and cause evolution of a species. Sometimes they are called open reading frame, sometimes they are called novel open reading frame, depending on various configurations, but I am not going to detail of it. This is what I have already discussed, so I am not going into details of it because we have only 1 hour. So long range regulatory elements are part of this so-called junk DNA, and cis regulatory element, they are also part of this junk DNA, and they control all epigenetic changes. So it is not an inert DNA on which environment works to form a epigenetic change. It is actually a very active DNA which allows or block environmental change to work on various gene processes. This is transcription where all the epigenetic regulatory processes start. This is RNA polymerase, which actually gets attached to various promoter and initiator sequences. And from then the transcription of mRNA starts. Now, nowadays, standing in 2023 January, we know that this promoter region is the most important part of epigenetic regulation in most areas. Various proteins and RNAs attach to this promoter region to start RNA polymerase action, and this promoter region is one of the master controls of whole epigenetic regulation. There is enhancer sequence in DNA, then activators attached to those area to form enantiosome. Then that enantiosome causes folding of DNA, and there is formation of mediator complex, RNA polymerase, and transcription starts. Be it step 1, be it step 2, or be it step 3. These 3 are the core areas where all the epigenetic processes that we know about happens. Yes, there are thousands of processes of epigenetic change, but these 3 are most known to humankind at this moment. That doesn't mean that other parts they are least important, but we know more about this area than the other areas. This is the full complex: initiator, downstream core promoter element, Tata box, and where the Tata box binding protein works, enhancer, activator, co-activator. This is the complex which, if not formed, then RNA polymerase would not work. And each of these elements, be it this mediator, be it this protein or this protein, or this protein which is actually more than 30 plus in number, these proteins, without them, no epigenetic changes would happen. Now, the second stage of epigenetics is splicing of messenger RNA and formation of the functional messenger RNA. There can be various forms of splicing. As you can see, various ways we can splice the messenger RNA and form various messenger RNA. I am not going into details of these processes because this would bore a normal clinician, because for clinicians this is not required. But this is the way mRNA splicing works, and all these ubiquitin associated proteins, they are one of the areas where various neurodegenerative processes start. Now, transport of this messenger RNA across nuclear pores into cytoplasm, that is nucleocytoplasmic transfer. This is another area where various epigenetic steps happen. This is the nucleocytoplasmic pore, this is the area, and various protein tracks to NXF1, P15, all these, they connect with pores to make it active and to make the mRNA transfer across the pore. This is a more clearer view of it, and see how many proteins are there which gets attached to mRNA and then transport the mRNA across the pore, and see how many proteins are there which gets attached to the pore and keep it open. I am showing these pictures to make you understand that there are various things apart from our so-called DNA methylation and other things. And these areas involve thousands of proteins. This is another view of it. And here we can see the various ways this nucleocytoplasmic transfer of mRNA gets hampered and produce or do not produce various defects. For example, this protein is actually one of the ways to control immune response and cell cycle, and possibly this is one of the proteins that is involved in various stress response. This is maintaining of pluripotency, TREX1, and if you remember, induced pluripotent stem cell require these protein complex to work. Anyway, I am not going into details of it. Again, not required at this stage. Maybe in future, after let's say 30 or more epigenetics discussion, we would be able to come to this area. Now, translation of messenger RNA into polypeptide. Again, this is not just a single process. The various component of ribosome assemble, then with it mRNA gets joined, then tRNA takes various amino acids to it, and each of these stage multiple cellular processes gets attached, and be it conversion of GTP or be it these areas, all of them, they are required for epigenetic control. This is more detailed view, but again, I promised that I would not go into detail and bore you. So ultimately, we come into formation of a polypeptide, and then how many changes that polypeptide can go for post-translational modification. There can be reversible or irreversible changes. If we do deamination or ethylation, then it would be irreversible. If we do proteolytic cleavage, then it is irreversible. Ubiquitination or hydroxylation or phosphorylation, they are reversible, as well as glycosylation. These are things that are reversible. And each of these steps involves all those processes that I have given a glimpse of. So there are hundreds and thousands of modifications of this process. For example, this is microRNA. How microRNA is formed, there is canonical pathway, non-canonical pathway. I'm not going into details of it, but these microRNAs goes into various messenger RNA, and through activation of slicer and dicer complexes, they actually degrade messenger RNA. They block messenger RNA. Someone is doing something. Please stop. Yes. Thank you for the consideration. So microRNA, by activating slicer and dicer complex, as well as activating various proteins to get attached to mRNA and various other ways, stop formation of proteins and modify these processes. Similarly, there is a RNA called circular RNA, which absorb microRNA and stop microRNA to degrade messenger RNA. So there are thousands of variations. These are the ways microRNA can affect messenger RNA. So what are the other non-coding RNAs that has a role in these epigenetic processes? So far, we know about 1,500 to 2,000 these non-coding RNAs. They can be small or long. They can be cis-regulatory, gene, intron, linear, circular. But again, these divisions are not much required for a clinician. Good to know, but not needed to know in details. These are the types which interferes with all the protein formation activity, and each of them can affect and do affect all the epigenetic functions. So then comes the endogenous retroviral element. One of the things which probably is very important in psychiatry, and again, actually a epigenetic process. Due to thousands and millions of years of evolution, all animal species on this earth has integrated various viruses in their genetic structure. Human being is not exception to it, and it also has various retroviral element as well as viral element inside our DNA. So far, what we have understood that 8 to 10% of our DNA is viral DNA integrated to our system. And these viral DNAs have a huge role in immune regulation, cell fate regulation, and regulation of various other processes, most of which are extension of immune function. So standing there, these endogenous retroviral DNA, they are important for formation of multiple sclerosis, as well as formation of so-called schizophrenia or rather chronic psychosis, formation of bipolar, formation of depression, and various things. But today we are not going to discuss about endogenous retroviral DNA. I would just give you a glimpse of it. So when we talk about gene environment interaction, which environment we are talking about? Because any animal has one external environment which comprises of physical environment and social environment of that animal, then internal environment. Internal environment can then subdivided into extracellular environment. Extracellular environment in turn is further changed into intracellular environment. This is external environment, this is internal environment, and this is cellular microenvironment. And each environment change is decreased whenever we go into deeper. Means any change in external environment would produce less change or very less change in internal environment, and any change in internal environment would cause very less change in cellular microenvironment, because each of internal environment and cellular microenvironment has their own regulatory mechanism, and body tries to keep the fluctuation to minimal level. So if we say external environment, then first level affected to the external environment is organ and tissue. Then that causes change in homeostatic and autonomic regulation, and then the central control flux. So 3 layer of filter. So standing there, if our external environment changes, that doesn't mean our internal environment change to the same degree and to that particular stimuli. If someone slaps me, then I would feel the slap, but my brain would process the slap differently. Then the impact of the slap would be different in different cells of my system, and ultimately it would differ from me to another person and things like that. This is extracellular matrix, and this is the intracellular area which we are talking about. This by environment, the environment which affects the epigenetics, we are talking about this area, this environment. Now this environment gets very less disturbed by something happening in external environment. As I was talking about the slap, if someone slaps me, then the change in the intracellular environment of my brain's neuron would not be different than if I have a fever in my body or I have an infection in my body. So the idea of the trauma of the all-pervasive trauma in psychiatry, that I had an trauma and so I have this problem, it has a basic fallacy. Brain and body doesn't understand whether the trauma comes from lifelong poverty, or someone is sexually abused at certain days, certain developmental phase, or someone had malnutrition or during certain developmental phase, or someone had some other environmental calamity during the developmental phase. So intracellular environment cannot differentiate between these. So the DNA methylation, histone methylation that the current research is finding is probably baseless and has little scientific validity of any abuse felt during developmental year. Those methylation, those acetylation, those changes can very well represent a life of hardship rather than a single sexual abuse or physical abuse. So this is what I was talking about. Intercellular environment won't know if the change is due to someone is slapped or have infection associated body change. The change in intercellular environment is always generalized rather than specific to a specific stimuli. This picture can make it understandable too. Be it environmental stress, be it oncogenic stress, be it growth factor signaling, they are together a form of input. That input is seen by writer, reader, and eraser type of mediator, and that cause RNA modification and that cause various changes. It doesn't matter whether it is environmental stress or growth factor signaling or something else for the cell. These are writer, eraser, and various things. I'm not going into details of them again. So standing there, let's take PTSD, the quit essential trauma and trauma-induced brain problem in human beings. Rather, epigenetics and PTSD has become an eponymous phenomena in psychiatry. Everything is now trauma, everything is now PTSD, and everything is now various other things. If I see a prescription and a medical file written by an American psychiatrist or Canadian psychiatrist or UK psychiatrist or Australian psychiatrist, I would find his or her note would always mention PTSD without understanding or without any validation of if there is any PTSD or not. PTSD is such eponymous in psychiatric nomenclature. So let's entangle the epigenetics of PTSD and thus try to understand what we learned so far, and let's apply our knowledge in making PTSD epigenetics simplified. So molecular biology of susceptibility to post-traumatic stress disorder, highlights of epigenetics and epigenomics. This was a paper that actually have looked into this. It is a very recent paper and it discussed how underlying susceptibility is more important in so-called PTSD. This study has found that there are various significant CPG changes in these areas. Problem is, there is no evidence that whether these changes are due to death or any trauma, or these changes are there, which resulted in those expression. Now, this is another cohort that looked into various military veterans, and they have found change in various CPG sites in their methylation. But again, the problem is nobody knows whether the methylation has happened due to being exposed to war trauma, due to being exposed to various conditions pre-war, or due to being just part of the genetic makeup of these people. We don't know. They just found that difference, but they never explained it. And it is not possible to even explain it, because it is not humanly possible at this moment to test a whole epigenome profile of every person who is getting recruited into military academy, and then doing their serial yearly epigenetic profile till, let's say, 10 or 15 years, and then compare it to another group of people who are not exposed to that. Then and then only you would be able to tell whether the combat trauma or combat experience or various exposures in military has caused these epigenetic changes or not. And so far, no data of that kind exists. Again, this is a paper which is again very recent, but they also stayed in the same way, that they tried to find some epigenetic change without understanding what that represents. But they have found out some biotype, and the G1 and G2 biotype, some of them actually responds to stress differently. So ultimately, this study, if someone is being able to follow it later, might give rise to some epigenetic and genetic understanding of epigenetics. Again, this is another paper, fairly recent. Am I visible?
Yes. Yes, you are.
Okay. Actually, what happened is I thought that connection got out. So anyway, so this is another study which looked at epigenome-wide association study of PTSD in US military veterans. They found various changes, but again, these changes are probably more about the risk-taking nature behavior, as well as various ways that makes these people more exposed to military rather than our signature of war trauma. These are peripheral blood PTSD signatures. Again, they have found various immune system changes as well as some global mutilation. But again, they have not been able to trace it to that trauma. They have just done a epigenetic test in some people who claims to have PTSD, and they found this methylation. So probably this and this methylation is involved in trauma without any replication of this study. We have to understand that none of the PTSD studies done so far has been consistently replicated. Same. So what is the epigenetic hypothesis of PTSD? Now, it can be same in all psychiatric problem rather than specific for PTSD. There is gene environmental interaction as we already described, and the underlying risk genes, they might be making the person vulnerable to various adverse events in environment, and in turn those adverse events in environment are being allowed by the same regulatory system to impact the various cell environment and ultimately change in various protein expression. And another thing would be various positive and negative factor in environment along with risk gene might modify the various ways of development and ultimately can lead to various pathological states. Problem is we at this moment don't have the technological advancement to find out whether these theories are right or wrong. So the idea of early life stress producing PTSD works like that. Okay. They say that stress causes change in FKBP protein and ultimately causes DNA methylation. DNA methylation causes change in BDNF, SKG, SDK1 and FKB5 and ultimately causes change in neurogenesis. Problem of this idea is, again, it doesn't take into account the underlying vulnerability profile and resilience profile of the person's gene regulatory mechanism. If the gene regulatory mechanism allows, then only this can happen. Otherwise, this cannot happen. So, in all these theories, the genetics and epigenetic regulation gets very visibly ignored. This is more a complete picture of the theories, and this is probably to some extent true that epigenetic processes and epigenetic regulation, they work side in side, and if the epigenetic processes allow the traumatic stress to affect the gene expression, then susceptibility compounded by genetic underlying genetic vulnerability would produce PTSD symptom. And PTSD symptom, seeing if a patient develop PTSD symptom, then there should be targeted epigenome-wide association study as well as targeted gene-wide association study to find these things out. So what are the epigenetic tests that we can do? This is trick-based assay, but I'm not going to detail of it. These are the standard tests that can be done. Although you can see that these
Excuse me, Dr. Bhaskar. Excuse me,
Yes.
Dr. Bhaskar. Another 6 minutes, please.
Yes, I have already almost ended.
Thank you.
These are the tests that are applied in various cancers, but all of them has their application in psychiatry and their application in various so-called trauma. Problem is, we would find something, but we wouldn't know whether that has caused anything or not. This is epigenetic clock. I am not going into details of it because epigenetic clock is another oak terminology at this moment rather than a true science. But I would just give you a trivia. This taste, remember this taste is actually the cause of Chris Hemsworth, who is actually the Thor in various Thor movies, taking his leave from movies because he has done one of these tests, and these tests show that his biological age is increasing. So he is trying to reverse that. Think how massive the propaganda is. So scientific critic. Epigenetics is literally million times bigger than just DNA methylation, histone modification, and few microRNA measurements. The epigenetic hypothesis of trauma forgot to understand that. Cell and DNA regulatory mechanism doesn't know what cause intercellular change. Sexual abuse, serious infection, both have similar environmental signature for cell. So how the epigeneticists come to conclusion that a set of DNA methylation, a tiny set of superficial epigenetic tests showing change means it's only and only due to their idealized trauma? This is something that epigenetics, people who are epigenetics and who are into epigenetics and trauma, they have to clarify how they understood that. And then the so far presentation clearly showed that changes in cellular and nuclear environment around DNA and impact on DNA regulation can only happen if the non-genetic regulatory DNA allowed that to happen. So epigenetics is actually an active process where environment is only allowed to change in function when regulatory elements has a defect in them or allow environment to affect gene function due to a synchronized gap in them. Environment in gene environment interaction again denotes internal environment, and that to intracellular and internuclear environment, which cannot for the psychosocial environment of psychiatry. What happens in psychosocial environment hardly have any say in internal or internuclear environment. So various tests that are now being promoted as epigenetic tests, they are leading to more confusion and they are not really true epigenetic tests. So with that, I end the overview of psychiatry epigenetics. I know it is a very huge presentation which I tried to compress. I don't know how much you understood. Just I hope I have been able to make you slightly curious and have instilled some amount of skepticism about the revered trauma and PTSD. If I have done that, then it is enough. Thank you.
Thank you,
I
Dr.
have done my
Bhaskar.
presentation.
Thank you, Dr. Bhaskar. Maybe we can stop screen sharing now.
Yes,
There are
yes,
a couple
yes.
of questions, Dr. Bhaskar.
Just a minute.
One of them is, how do you will you take the questions now, Dr. Bhaskar?
Yes, yes, yes.
So
Just a minute, let me stop the screen share. Yes, screen sharing off.
Yes, it's off, yes. So how do you conceptualize epigenetics as? Is it an upward causation or a downward causation from the level of thoughts to atomic level?
It is from not the level of thought. It is thought is just various EPSPs in neuronal cell. They sometimes sum up and produce recognizable action. Most of the time, they don't sum up and just stay as microfracturation in the electric grid of brain. They doesn't change any gene function or other thing. Rather, they are expression of gene changes. So thought cannot change any epigenetic function. So epigenetic function is always at the level of epigenome, so at the level of various regulatory DNA element and intercellular environment. So I don't know whether whatever you want to call it, bottom up or bottom down, doesn't matter.
So then the next question is in a way kind of linked to the first one. It says we can explain acute symptomatic manifestation of PTSD or chronic condition of PTSD on epigenetic explanations.
How?
That's what he wants to know. How
Can
do we
we?
explain?
Yeah, can we? And
Can we?
this epigenetic manifestations, do they explain the acute PTSD or chronic PTSD?
Epigenetic changes, the changes which may or may not be related to trauma at this moment doesn't say anything about whether they are specific to PTSD. That is the number one problem. Let me first delineate the problems with epigenetics and PTSD. Number one, the changes we have found so far is not specific to epigen trauma. If I had more time, let's say 3 to 4 hours, I would have shown you by precise tracing of the epigenetic changes that has been so far found in various studies. And I have shown you the tracing goes to immune function, tracing goes to various innate immunity and adaptive immunity, tracing goes to complement function and other things. They doesn't link anything to trauma and other things. Anyway, so this is number one problem. Number two problem is acute PTSD is nothing but an acute post-traumatic response. That is non-specific. Someone slaps you, your near ones dies, you had a negative event, all the time you would show the same reaction. The reaction of a human being who has faced a negative event. Now that you can call it grief, you can call it acute PTSD, you can call it various other things. Doesn't matter. It's just different semantics. Now, whether why that is more in some people, why that is less in some people, maybe due to those person who have it more have in their brain various genetics which doesn't make their brain work properly. Those genetic changes might or might not be aggravated by various memories, various life experiences they have suffered, and that might have led to some epigenetic changes, and that is how things work. So be it acute PTSD or be it chronic PTSD, both the areas have some link to epigenetics, but epigenetics is not the whole and soul of PTSD. If we go only via epigenetics, we are awfully short at this moment on information and infrastructure to say much about PTSD. So PTSD is much more than just epigenetics. Next question.
Well,
Hello.
I entirely agree with you, Bhaskar, that everything cannot be explained just on the basis of a particular type of trauma, but you do agree that trauma can cause changes in DNA methylation and other epigenetic processes, which may have differential results.
Ma'am, I don't agree
in
to
people.
that. I don't agree to that. I am not
I think
agreeing to the fact.
that are some of the questions.
Yes, I am not saying that trauma is causing a particular epigenetic change, because it is not possible
Right.
for
No,
a
I agree
cell
with
to
you.
understand.
Agreed,
Yes.
agreed. That's what I'm saying, that
Hmm.
it one specific trauma may not cause one specific disorder,
Yes.
but
Hmm.
broadly speaking, if there is trauma and that
Hmm.
continues for some time, for instance,
Yes.
malnutrition or continuing child sexual abuse,
Yes.
then it does cause changes in the epigenome, and that eventually causes changes in the entire bodily systems, both physically as well as physiologically as well as psychologically. And that leads to some of the questions here, which say that, does psychotherapy change epigenetics, or does it
No.
influence epigenetics? Does
No.
there was another question: do medicines cause epigenetic changes? Is diagnosis now an epigenetic matter? These are some of the questions which are there in the chat box. You can see.
Yes, I am seeing it. Someone named Kumar has questioned, if thought doesn't affect epigenetics, how psychotherapy works in bringing in changes in mood and other cognitive. It doesn't work via epigenetics. Psychotherapy doesn't at all work via epigenetics. Thoughts doesn't affect epigenetics. What happens is, in psychotherapy, we employ, or rather I would say those who are doing psychotherapy employ two pathways. Number one, the prefrontal cortex is troubled in these people, and it is not being able to find solution for a lot of life problem. There, the therapist help the prefrontal cortex in reducing its burden by providing various alternative paths of management. That is number one way how psychotherapy works. Number two pathway is very interesting pathway. Reward pathway is probably one of the strongest pathway of human beings as well as any animal being. Reward pathway is also the pathway of hope. And by structurizing and systematizing a person hope response, psychotherapy causes changes in mood and other cognitive function. That has nothing to do with epigenetics. Please don't insult epigenetics. Then comes, what about the epigenetic aspect of intent? Exactly. Maternal and child environmental changes and its epigenetic is a fascinating thing. Fascinating thing means mother has its own immune system and its nutritional system that gives an input. Then mother's various other body states gives an input, and those inputs are dealt by baby's body in the system, then baby's cell system, then baby's intranuclear environment. And that again causes change in the form of feto-maternal, basically transmission. Means fetal cell are being transmitted to mother's body, mother's cells are being transmitted to fetal body, and that again cause a level of epigenetic complexity that is unique to pregnancy. And ultimately, feto-maternal epigenetic environment is one of the most interesting part of epigenetics that we know. I would be happy if I can discuss about it much more, but I have a train to catch, and this discussion would last for a few days. So if you are interested, let's start our discussion somewhere else. But at this moment, this space is pretty small for this discussion. Then someone has asked, or whether all medicines in all specialties do alter epigenetics. Yes, they alter. Every medicine in human pharmacopoeia has epigenetic potential. How medicine changes epigenetic? Medicine either changes DNA methyl transferase activity or histone methyl transferase activity or histone deacetylase activity, or it can changes a protein's potential to bind with methylated histone or acetylated histone or phosphorylated histone. It can also causes phosphorylation of broken DNA fragments and ultimately leads to healing of the DNA and DNA repair and other things. There are thousands of ways the medicines can cause epigenetic changes. But if we, I can give you a broad scheme. But if you ask me that Bhaskar, whether this medicine cause this, and this epigenetic change, I would say I don't know, because we know the general schema. We don't know whether any particular medicine is working any particular environmental way or not. Sorry, but a particular epigenetic way or not. Now another person has asked, can we assess level of susceptibility to trauma, epigenetics of a person? Impossible. You are talking about, I don't know how many of you have seen the movie Gattaca, but you are talking about level of surveillance of that area. You have to do whole genome sequencing and whole epigenome sequencing of every person born in every part of world to understand that. It is impossible at this stage. Maybe in future, but not presently. Now, another question, does present psychiatric diagnosis has any relevance at epigenetic level? Present psychiatric diagnosis has no relevance at any medical level. They are just for making the insurance company's claim possible, because no insurance company is going to give you reimbursement to your patient if you say the patient has this and this symptom. So we need to give them some names. That is why we need DSM and ICD. But at the level of epigenetics, at the level of epitranscriptomic, at the level of various things, these diagnoses doesn't matter. So again, transgenerational trauma. Transgenerational trauma, first, we have to understand that transgenerational trauma is of multiple things at multiple animal level. For a nematode, transgenerational trauma is easily visible. If you breed a dark nematode in a light environment for quite some time, that environment would change the nematode's color. Direct effect of transgenerational trauma or transgenerational changes. But for complex animals like human being or elephants or blue whales or any whales, their transgenerational trauma is hard to measure at this point. Probably it happens because biological principle doesn't differ based on the level and complexity of an animal. But for large complex animal, the way to measure would be very difficult, and the effects of that would be very subtle. There was a famous study in epigenetics, the Dutch hunger study. During World War II, the Dutch people were starved by Nazis, and that led to various problems in their offsprings. This is one of the examples that we used to give for transgenerational epigenetic changes. But now we know that it is not transgenerational actually, because at that time of that drought, those babies were inside the mother's womb. So they were already affected to various malnutritional problems. So being exposed to those malnutritional problems, they developed with various changes in their body, and that probably led to various things. So it is mostly intergenerational trauma rather than transgenerational trauma. True transgenerational trauma would be grandmother being affected by something that would be passed to mother, that would be passed to granddaughter, and that granddaughter would produce a child who would show the same problem. That would be true transgenerational trauma. At this moment, we don't have any evidence for it. So environmental
So
modification, yes.
yeah, please go ahead. Please complete what you were saying.
Yes, one person has asked that whether environmental modification would be given same importance to treatment as medicine. Environmental modification has some role like healthy living lifestyle, doing active work. That is endorsed by every specialty. But that doesn't mean that we would be giving it more role than auxiliary role. They cannot be mainstay of treatment in any area. So anything else? Anyone, any other question?
Well, at some point of time, we do have to call a halt to the questions.
Yes.
Because so I'll
Yes,
give
I have to run
Dr.
for.
Yeah.
I have to run again.
Yeah. Okay. So basically, I would say epigenetics is still evolving, and as is true for all other branches of psychiatry, we do need rigorously conducted long-term studies. There's absolutely no doubt about that. Be able to prove or disprove a particular theory about epigenetics. I hope you agree with that. And on that note of agreement, I think I will hand over the mic to Dr. Vihang Bhaiya, who will, in his inimitable style, will conclude the meeting.
Well,
Yes.
this is one of those events where I'm completely speechless. There's a lot of food for thought. I'm still trying to digest some of the terms, leave alone the explanations. But I'm sure over a period of time, even a person at my age would understand a little bit of what Bhaskar was saying. Bhaskar, thank you very much for your in-depth study. You tried to make it as simple as possible. And
did I make it too complex?
no, you did not. The topic itself is complex, so you can't make it more complex. But you tried to explain to some of, I was talking to some of my colleagues and they seem to have understood much more than I did. But well, there are some things that we understand, and let's hope we have more of these meetings to understand it better. Thank you very much. Thank you. Bye.
Thank you so much, respected chairpersons, Dr. Smita Deshpande and Dr. Vihang Bhaiya. And thank you so much, Bhaskar. I know you have a train to catch and you've still been here with us and shed light on this topic. We have a lot of psychiatrists today joining us from Sri Lanka as well. And they were very happy to kind of know that this is a free access web series that they can join in. So welcome to our colleagues from there. And it's been an exhaustive session with a lot of explanations, and that leaves us with many more questions. And I thank Dr. Aleem Siddiqui for being with us and as the welcoming committee as well as the editor pushing for our journal. Thank you so much. And at this point, it is a pleasure to introduce to you guys and invite Dr. Prasanjit Ghosh from Silchar. He is the honorary treasurer of Indian Association of Biological Psychiatry, and being the money man pretty much allows us to do what we've been doing. So thank you so much, Dr. Ghosh, for joining us, and I would request you to share a few words.
Thank you so much, Rukseda. I, on behalf of Indian Association of Biological Psychiatry, convey our appreciation and best wishes to Dr. Bhaskar for accepting our invitation to become tonight's speaker. And he delivered a very thought-provoking lecture on a still evolving concept. I also thank our chairperson, Dr. Smita Madam and Dr. Vihang Bhaiya sir, for nicely handling the session. And I also thank all our participants for making tonight's CME very interesting and posting so much questions, and still some questions are unanswered. And also thanks go to our dynamic secretary for continuing and conceptualizing actually this academic series. I thank our academic partner, and also I thank our editor, Dr. Alim Siddiqui, for doing the initial part of tonight's event. And also thank goes to our office assistant, Ms. Amita, for the logistic support. And wishing you all a very new year 2020-23. And that's all. Goodbye and good night.
Good
I'd
night,
like to
everyone.
also remind, I don't know if Ruxida remembers, the IABP channel is there on YouTube, and I suppose we will be able to see these slides whenever
Yes,
we want,
yes,
if we
yes,
subscribe
ma'am.
to this channel.
Yes,
Yes,
ma'am.
yes.
Thank you so much, ma'am. You stole the words right out of my mouth. We upload these because, like I said, it's a free access program. So we also upload it on YouTube and share it. So for better and more such webinars and sessions, I would request everybody to like, subscribe, and share, and also press the bell icon so that we can keep continuing to do our efforts in biological psychiatry. Thank you so much, Dr. Vahia, again, and thank you so much, Smita, ma'am. I know you're traveling and you've come to Bombay for a conference, and it's really sweet of you guys to always be there for us and for biological psychiatry. And with that, I wish everybody good night. Take care of yourself, and those of you who haven't had dinner yet, please go feed yourself. Bye. Good night.